Now we have the Young Scientist Award, and we have 10 contenders for that. So judges, this is a different session, a part of the same session, but different award. We have a Young Scientist Award for one among these 10, and we have one Best Paper of Session where the 4 keynote speakers have been attributed. Are we ready? Can we start with our pre-paper presentations? Yeah. Now, the rules of the game, children, who's starting first? Come. Ajo.
The Proteo.net from Anaga Setlur.
Dr. Anaga.
And she is from AIML Biotechnology track.
And what's her paper?
It's Proteo.net, novel GNN-based ligand binding affinities
Ready?
prediction.
Anaga, ready? Okay.
Just 3 minutes.
Anaga, we are going to try and put up the clock timer over here. You're given only 3 minutes. I'm having Anirudh stand next to you. So he'll go tap, tap on your desk. Okay? Come. This session is actually meant for all you students, only so that you can learn to give the 30-second elevator speech. We've done too much work. There's too much work that you need to put into practice. You can't. It's not humanly possible to tell all your experiences, all your PhDs, all your startups. You need to be able to condense that. And that condensing is what we are trying to teach our young scientists. So they have time. Judges, time is of essence here. Yes, Anaga, 3 minutes, start.
Good afternoon, everyone. I am Anagha, and today I'll be talking about Proteon.net, which is a novel GNN-based ligand-binding affinity predictor architecture. And this is done via SMILES, and we've done this for 3 important organs of the human body: liver, kidney, and brain. So just to briefly talk about the background, so we know that traditional QSER models basically predict how the chemical structures relate to its biological activity. This basically helps in understanding and estimating protein-ligand interactions. But they, however, struggle to fully capture all of the interactions that are there, especially the ones that are complex. That is why there is a need for an integrated platform, and that is why the problem statement is to directly predict the ligand binding affinity to the 3 organs that we have selected using a structural approach. And the solution is to use graphical neural network based architectures. So the main objectives were, one is to generate and validate the QSAR models, to design and evaluate the GNN architectures, and to develop a user-friendly real-time docking score or binding affinity predictor. So this slide talks about the overall methodology as well as the tools and techniques. So we have 3 methods, that is data collection, pre-processing, as well as building of the model. In data collection, we collect and select specific proteins from all the 3 organs. Then we have building the ligand library, docking, as well as performing QSAR models. In our study, we performed 300 plus QSAR models and built them. Then we pre-process the data by initial data set preparation, followed by model building. That is, we built 2 models. One is GCN, Graph Convolution Networks, and a hybrid version of the first one. Then we deployed it on Streamlit. So these are our results. This basically shows a sample representation of how our QSAR plots look like, which is seen in the graph on top, as well as we have the distribution plot also. So these are the 2 models which we used. One is the simple GCN model, wherein the graphs are correlated to the target variable, and in this case, it is the docking score or the binding affinity. And we have model 2, wherein one additional attention mechanism is taken into consideration, which is the positional encoding. So this slide talks about our initial data set, what were the small molecules and how we kept it all and correlated them. Then we did a feature representation. So since this is a preliminary model, we took only 5 features to represent them, and these are the 5 features which were taken. And the docking score app was then deployed on Streamlit, and the application and link is also given here. So the application somewhat looks like that, and it gives users 3 options. One is to select the organ in the human body, then to also select the protein which they want to check their docking score against, and also to build a model. So they can select either model 1, which is the GCN, or model 2, which is a better version of GCN and gives a better accuracy. Yeah. So these are the
Time's
two
up.
publications which we have got from this study. Thank you.
Thank you, Anika. Thank you. Thank you very much. I'm not going to ask questions now because we're running a little late. We'll go to questions at the end of the session. Dr. Advait, please come. Anirudh, please read out what
So
is
we have the
Advait.
second poster presentation under AIML in Biotechnology track by Advait V. The title is Transportation Networks Inspired by Growth and Evolution of Slime Molds.
The timer is on a PIP, so you can see it up there. Okay.
Yes, ma'am.
Thank you. Ready, Advait? You looking so...
Okay, so I'm going to talk about bio-inspired transport network systems. So we, as Bangaloreans, have been stuck in traffic forever and ever, and we need to get around quick and fast. How many here have been stuck in traffic within the past 24 hours? Quite a few. There we go. So the problem statement is tackling this problem that we have. Okay, so introduction of so I have a solution to this problem that we have, that is to tackle the traffic problem and the roads of Bangalore or any other place in India. So before going to the solution, I'll just give you a brief history about all these, where we took the inspiration from. We took the inspiration from an organism called Fusarium polycephalum. That's basically a slime mold, and it has existed for about 600 million years, and we have existed for a very minute time according to that. And we honestly, something that has survived for the past 5 great extinctions ought to know something better than us, that is to survive. And the main objectives of this problem statement that we have is how to transport properly, how to distribute population, traffic management, and water management. So after that, these are the materials used, not that important though. Okay, so coming to the main problem, what are we using to solve the problem? The traveling salesperson problem is a very common thing in computer science, where we talk about how a traveling salesperson has to go from one house to another house constantly just to deliver substances such as newspaper or anything. And this slime mold encompasses all of that information and takes it and solves it within few seconds. Okay, this is the physical experimentation that we did in our RVC biotechnology lab with the help of Dr. Nagashri ma'am. At the end of day 7, there you can see, although the straight path would have been the most efficient, shortest path, it took this path, showing that it's the most efficient path because that would cause a lot less traffic for the slime mold to grow. Okay, so here are the basic applications of it. We'll have a better, efficient city designment, and then we can control movement, traffic organization, and coming to the medical part of it, we can integrate nanobiotechnology, AI, and this, so that we can solve problems such as catheterization that can be completely automated. We wouldn't need doctors to do that. We could have the slime mold grow within another place so that it can do catheterization right there. Okay, so this is the algorithm that we made based on the physical experimentation. So we have 2 algorithms. That's a map simulation at the first, where you can just pin out any points that you want, and then it'll give you the most efficient path to go from one place to another place. As you can see in those clustered areas, those are the population dense areas, it'll give you the most efficient path. The second one is a map finding path, where it'll give you the most efficient path to go from point A to point B. So if you scan that QR code, you can go to our website and try this out for yourself right now and anytime you want. So, okay, with that, this is the outcome, and thank you.
Good job, Advait. I'd like to call Falkia Taranum.
So now we're moving on to the
And,
second track, which is healthcare. So in that, as ma'am just called on, we have Falkia Taranum, and their title is Steady on Cytokine Production by T lymphocytes triggered
And
by
you guys are doing
uraciols
wonderful.
from rest tox tinctures.
Superb job that you're keeping to your time. Excellent. Your stop clock is there. Okay?
A very good afternoon, everyone. I'm Falka Taranum. We recently conducted a study on cytokine productions by T lymphocytes triggered by urushiol from rust tincture. Urushiol are basically...
What happened?
Can you please move to the next slide?
Please, yeah. Can we restart?
Timer again.
Change or you guys...
Is this your talk?
Yeah.
Okay. So we'll start the timer again. Let's go.
A very good afternoon, everyone. I'm Falka Taranum. We recently conducted a study on cytokine production by T lymphocytes triggered by urushiol from Rustock tincture. Urushiol are basically plant-derived catechols found in Anacardiaceae family. They are known for causing acute contact dermatitis, a type 4 hypersensitivity reaction. They are known for causing symptoms like skin lesions, inflammations, and swelling. The purpose of our study was to investigate the anti-inflammatory properties of rust stocks and their impact on immune cells, and also their ability to modulate to modulate their cytokine production and reducing the inflammation. Moving on to methodology, we used two commercially available rust stocks from Schwabe and SBL and cashew nut shell extract, which we used as a substitute for commercially available urushiol. We also did both qualitative and quantitative estimation of polyphenolics using several advanced techniques such as GCMS and TLC. We also did cytokine assay, and also we used blood samples from both naive individuals and those allergic to cashew nuts to check the CRP levels using turbidometric assay. We also developed a novel method of culturing T lymphocytes without the use of growth mediums like RPMI or fetal calf serum, therefore reducing the cost of cell culture. So these are the results. The phytochemical analysis and TLC showed several bioactive phytochemicals, in specifically phenolics and flavonoids. So the GCMS analysis showed several compounds that can cause irritation and several compounds that can cause that showed anti-inflammatory properties. So the absence of urushiol, it also showed that there was no detection in urushiols because there might be some breakdown by an enzyme called lacase, which during the tincture preparation. We also saw there was no changes in CRP levels in both the blood cultures. It might be because the CRP might be CRP might not be the inflammatory marker in whole blood inflammatory marker, or it can be that anti-inflammatory compounds present in the tincture could have impact on them. So to conclude, our research has shed a light on phytochemical analysis of rust stocks tincture and their impact on immune cells. We also saw there was no role of CRP in CRP and yeah, next. Thank you. This project was funded by KSUST.
Thank you so much. So next, moving on to the second participant under healthcare, I invite Hima Bindu. Her title is Targeting Friedreich Ataxia, a Sustainable Path to Safer and Smarter Therapeutics through Integrated Docking and Toxicology.
Good afternoon, everyone. So our study mainly focuses on Friedreich ataxia. So the topic is targeting the Friedreich ataxia, a sustainable path to safer and smarter therapeutics through integrated docking and toxicology. So now let me brief you about what is Friedreich ataxia disease. So it is the autosomal recessive neurodegenerative disease which is caused mainly in the age, like the symptoms are caused in the age of 5 to 10, and the disease can be caused in 25 years old. So in this disease, it is caused due to the mutation which occurs in the FXN gene because of GAA trinucleotide sequence which occurs in the intron of the FXN gene, which blocks the transcription process, due to which the frataxin protein is not being produced. If frataxin protein is not being produced, it causes a major problem for the body because it is a mitochondrial protein. So if no frataxin is being produced, it incorporates iron and sulfur ions in the mitochondria, due to which it causes free ionic iron accumulation, due to which the ATP production does not occur in the cell and the cell starts dying. The other way is because of the free ionic iron accumulation, the free radical formation takes place in the body, due to which oxidative damage occurs in the DNA and the proteins, due to which the cell dies. So because of this disease, we did the research. We found that till now, there is no such treatment which directly cures the root cause of the disease. That is, it doesn't solve the problem of epigenetic silencing of this gene. See, the treatments which have been done till now are only focusing on the symptoms which have been caused by the disease and reduce them. But we have found that the therapeutics which have found, one of the example is Omevelixolone, which comes from the Skyclaris company, which mainly focuses on symptoms, but it has caused many side effects to the body, due to which it is not solving the problem of the disease. The second challenges which is faced by the drugs are because of the complexity of the mechanism of this disease, as it is a mitochondrial protein, many biochemical pathways have been affected to the body, due to which the person who is suffering from this can also have heart diseases and can die. And the third is the therapies which have been found have major toxic problems which have been causing a side effect to the body. So our team has focused on finding a drug which has more efficiency than the present drugs and has less toxicology. So the methods which we have used is finding a novel drug discovery using predictive modeling, that is computational modeling. So in this, we have used a platform known as Galaxy EU, which gives a environment and which gives the environment to perform the docking. So here we have taken these several compounds.
Thank you. Okay, now we move to the second participant under genetics. So we have genome-wide identification, functional characterization of protein phosphatase 2A by Nitya Fernandez.
Good afternoon, one and all. I'm Sister Nitya Fernandez. I'm here to present my paper title, Genome-Wide Identification, Functional Characterization of PP2A gene family and its role under abiotic stress in Cicer-Aratinum. Well, before we go into the study, I would like to state my problem statement. Despite the role of Arabidopsis PP2A genes have been well studied, and it's well documented, but the role of PP2A genes in Cicer-Aratinum is not been well known, and hence we have come with this objective saying that identification and functional characterization of PP2A genes and its role in abiotic stress in Cicer-Aratinum using genome-wide identification studies. So, well, here are few of my findings of my studies. First one, to study about the evolutionary relationship of PP2A genes of chickpea, with different plant species containing these PP2A genes, a phylogenetic analysis was conducted using MEGA software. Furthermore, to evaluate the structural characterization and the diversity of PP2A in chickpea, a correlative study of motifs from PP2A peptides of Arabidopsis thaliana and Cicer-Aratinum was conducted using the meme suite software. There are 21 genes of PP2A chickpea genes which are widely distributed across the 7 chromosomes as it is represented here. Next. The cis, that is care, cis-acting regulatory elements analysis helps in defining the responses to various stress factors by the plant. Next. As we move further, the RNA data was analyzed from which the potential role of PP2A chickpea in response to drought and stress factors were obtained, and the heat map was generated using Heat Mapper server online. The green color represents the upregulation in the drought stress as well as in the salt stress. Next. The structures of PP2A proteins were obtained from TR Rosetta, both abiotic, Arabidopsis thaliana and Caesaratinum, which were superimposed by using pymol, which was then, which showed that PP2A, that is the A subunit proteins of chickpea showed similarity with the PP2A subunit of Arabidopsis thaliana. Next. To identify the structural characteristics of PP2As, the exon and the intron structure of the genes were analyzed using the gene structure display server. Next. Concluding to my research that I have done, firstly, we identified there are 21 PP2A genes in Caesaratinum. Expression analysis showed that 5 genes, both in drought and salt stress.
Time's up.
Thank you.
Okay, now I'd like to call upon Mela Sudha with their title being Multi-Ligand Docking. This is also under genetics track. Just tell next.
Are you starting? Good afternoon, everyone. I'm Merla Sudha from Jayanagar University. Today, I'm going to present my art study on multi-ligand simultaneous docking of benazepril and captopril reveals enhanced affinity and synergistic interactions with ACE. ACE is angiotensin converting enzyme which converts angiotensin 1 to angiotensin 2. And angiotensin 2 in turn induces constriction of the blood vessels, which will in turn it'll constrict the blood vessels, which will in turn increase the blood pressure. So now there are 2 inhibitors which are FDA approved, which are benazepril and captopril, which are often prescribed by doctors for patients who suffer from hypertension. So these inhibitors are known to inhibit ACE. Now we have performed single docking and multi-ligand simultaneous docking. So in our study, we have performed single docking and observed that in the 2D interactions, there are unfavorable bonds with these both inhibitors. And when we observe the binding affinities, the binding affinity of the single dockings are less compared to our MLSG study. So MLSG is multi-ligand simultaneous docking where you dock 2 ligands at the same time. So in this case, we plan on introducing the concept of combination therapy where we want to introduce both the ligands together. So here we observe an inter-ligand interaction which conveys that both the ligands complement each other and show synergy. So along with introducing the concept of combination therapy, we also explored the influence of the polymorphisms associated with ACE. So why we are looking at polymorphisms is because the genetic variations in each protein might lead to a functional or a structural change which might lead to the change in the affinity of the drug. So for example, if any individual with certain polymorphisms, if they have the polymorphism, they show a different response to the same drug. So in this way, if we understand the polymorphisms, we can tailor a specific personalized medicine so that even that individual can have a special treatment. So in the methodology, we have taken the amino acid sequence, homology modeled it, and then we have performed the single docking and multi-ligand simultaneous docking. So these are the results that we have after we have performed docking with a polymorphic sequence. So as you can see in our single docking, we observe unfavorable bond with the first inhibitor, and the figure 6 represents the multi-ligand simultaneous docking. Now, so to conclude, we want to propose the combination therapy with both the ligands, and also we want to study the polymorphisms in each individual so we can come up with a personalized therapy even for people who are not used to the standard medication available.
Thank you. Now we're going to move to the next track, that's nanobiotechnology. So I call upon Shruti A. Rao with her title being Bacillus, Synthesized Silver Nanoparticles and its Efficacy in Controlling AIDS aegypti.
Good afternoon, all. I'm Shruti Erao, and I'm here today to present my team's work that basically deals with vector control, that is Aedes aegypti. We know there is a dengue outbreak every year, and around 390 million infections are reported every year at a global scale, and it's very important that we come up with certain green methods that can be used for the control of vectors, especially mosquito vectors like Aedes aegypti, which transmit dengue virus. Me and my team, we came up with a green method that is the microbial synthesis of silver nanoparticles. They are highly efficient in vector control. They kill mosquitoes. So our methodology goes like this. We identified and isolated our bacteria. It is a soil bacterium. We uploaded on NCBI and we got the accession number. It is Bacillus velezensis. Following that, we optimized conditions for the growth of our bacteria and subjected them to, so the growth was going good, and then we subjected them to stress, the bacteria. So they started secreting secondary metabolites, which are very, very crucial for the reduction of metal ions to metal nanoparticles. We subjected the secondary metabolites that we got to, we treated it with silver nitrate solution. They got reduced to silver nanoparticles, and it was visually indicated through a deep red coloration. To further characterize our silver nanoparticles, we conducted, so this is our culture broth, and the accession number is given here. We conducted UV visible spectroscopy, and the peak showed that there was formation of silver nanoparticles, followed by Fourier transform infrared spectroscopy, that is FTIR tests, to know that it was an extracellular synthesis of silver nanoparticles. It was followed by XRD to determine the size of our silver nanoparticles. It was around 56 to 59 nanometer scale. It was followed by SEM imaging, that is scanning electron microscopy, and EDAX, energy dispersive X-ray. We found out that the metal is found in its elemental composition, and also that our silver nanoparticles are cylindrical in shape, and they have a smooth surface. Following that, for the application part, we conducted many lethal concentrations tests and mortality percentage tests using probit analysis, and we found out that the larvae of Aedes aegypti, which were rare, they were sourced from NCDC, National Center of Disease Control. They were very much susceptible to our product, that is silver nanoparticles, especially as you can see in the graph, the second instar larvae.
Time's up.
Yeah, they were, thank you.
Thank you so much. Now, moving on to the next person under nanobiotechnology, Farhana. Their title is a comparative study of layered double hydroxide in basic nanoparticles as a potential catalyst in the conversion of microbial lipids into biodiesel.
Good afternoon, everyone. Myself, Farhana from MSc Biotech from NMKRV College, and I'm here to give a presentation on a topic, a comparative study of layered double hydroxide and basic nanoparticles as a potential catalyst in conversion of microbial lipids into biodiesel. In our research, our basically research consists of comparison of two types of nanocatalysts based upon their catalytic performance. Traditional biodiesel primarily utilizes vegetable oil, but recent studies have been shifted towards microbial oils. The transesterification process is carried out using catalysts that can be nanocatalyst or LDH. LDH have shown to be promising, and also with that of nanocatalysts have shown to be promising result by enhancing the efficiency, reducing the catalyst weight, lowering the reaction temperature, and also increasing the reaction speed. Moving on to the next one, that is materials and methods. First one was isolation of the bacteria, and which was where the cultures were extracted using blind Dyer method, and estimation of the total lipids were done using TLC and SPV method. Then synthesis and characterization of nanocatalyst and LTH was simultaneously performed, where green synthesis of nanoparticles such as we have used zinc oxide and magnesium oxide using the extract of stem and leaf of Colocasia esculenta. Parallelly, chemical synthesis of LDH was also performed using co-precipitation method. As these nanocatalysts were synthesized along with the microbial lipids, transesterification was carried out, which was characterized using FTIR and GCMS. Moving on to the results, figure 1 shows the oil red O stain bacteria. The figure 2 shows the profiling of the lipid using TLC. Figure 3 shows the estimation of the lipids, that is by sulfophosphovalenine method. Figure 4 shows the XRD patterns of the crystal size of the ZNO, MgO and LDH. Then figure 5 is the SEM analysis, where we determine the structural morphology of these nanomaterials. Figure 6 is the EDAX. Moving on towards this figure 7, that is FTIR, where we can see the peaks which determine the fatty acid methyl esters in the biodiesel. Moving on towards the conclusion, our study identified and characterized the obtained fatty acid methyl esters with that of using catalyst was determined to be in zinc oxide it was 35, and in magnesium oxide it was 45, and with that of LDH it was 25. Hence the 21%. Hence the obtained fames were comparatively high with magnesium oxide than that of zinc oxide and LDH. Since by comparing these emerging catalysts, we can, as you can say, this research contributes valuable insights in refining biodiesel production techniques, ultimately supporting the greener approach.
Thank you. Okay, now moving on to the next track, sustainable agriculture. So I would like to call upon Bharat Kumar. His title is Phenotypical and Genotypical Identification of Wood Decay Fungi from Shimoga Patches of Western Ghats, Karnataka.
Good afternoon, everyone. I am Bharat Kumar S., Research Scholar, St. Joseph University, Bangalore. So my topic is phenotypical and genotypical identification of wood decay fungi from Shimoga patches of Western Ghats, Karnataka, India. So here, this is my survey area, Shimoga. So here I have collected mainly 20 wood decay sample, and majorly I have selected sawmills and forest depot. So in the 20 samples, I come up with 2 novel species of Ganoderma. So along 20 species, majority of the most dominant species are Polyporaceae and Hymenocetaceae. Coming to the introduction, so there is a difference between mushrooms and fungi. So these wood decay fungi are conch, bracket-like structure compared to mushroom. So these mushrooms are smooth in structure. Coming to the color and texture, based on color and texture also, we can easily identify which is wood decay fungi and mushrooms. And based on the location also, we can identify. These are my objectives of research: documentation and diversity of wood degrading fungi and biocontrol. So this is the problem statement. These wood decay fungi, these are the major threats to the timber yielding plant or valuable plants. So I am going to control these fungi, and these are the tools and techniques which I have used: microscope, camera lucida, and Mega software to build the phylogenetic analysis. And this is the study area, Shimoga district. These are the collection. So I'll collect major sawmill and forest depots of the Chikmagalur district, Shimoga district. And these are the identification of wood decay fungi with the help of macroscopic and microscopic character. And this is the isolation technique. After the isolation, I have sent to the Rajiv Gandhi Biotechnology Lab for the molecular identification. So here result, most of the fungi I have found majority like a living tree compared to dead tree fungi. And these are the table which I have found the different type and host of the fungi. And this is the taxonomical work. So in that Ganoderma lucidum, Ganoderma sessile, and Adspermum, Asterales, Gibbosum, Xyleria, commonly called dead man's finger, Daldenia, Xyleria, Acrosporulus, and Daldenia, another species. And this is my novel species, Ganoderma tectopravine. Ganoderma is the genus. Tecto is I have found associated with the Tectona grandis. Pravine is my guide name. And this is the gene bank accession number. And this is the Ganoderma switanium. So these fungi I have found switanium mahogany plant. And this is the gene bank accession number. And this is the phylogenetic tree. So these are the different. Thank you.
Okay, next person in sustainable agriculture, I would like to call upon Leah Merin. The title is Valorization of Chicken Feather Waste Using Keratinolytic Bacteria for Sustainable Plant Growth and Waste Management.
What if one day you open the doors of your house and see a pile of chicken feather waste just in front of you? This situation is not far from a near future if in case we fail to manage the accumulation of chicken feather waste. I am Leah Merin Stanley, research scholar from Christ University. And with immense pleasure, I would like to introduce the title of today's presentation, which mainly includes valorization of chicken feather waste. So these are the screenshots representing how day-to-day activities of our life are being interrupted by accumulation of chicken feather waste. So the main protein demand of growing population is met by chicken meat. It is estimated that in the coming year, by 2050, the amount of poultry production will exceed 27 billion metric ton. This exceeding figure is of a great concern when it comes to the amount of waste which is getting released into the environment. But with the help of bacteria, we can degrade chicken feather and thereby we can reduce the load of chicken feather as well as create beneficial byproducts from chicken feather. So with this aim, I have formulated an experiment which mainly include isolation of feather degrading bacteria, which is called as keratinolytic bacteria. And these are the results: collection and degradation of chicken feather using bacteria, and finally taking it for plant growth studies. So for characterization, we have used several techniques to confirm feather degradation and find out the nutrient value of the solution. This is the result. The first picture represents the isolated bacteria, control media with the intact chicken feather, degraded feather, feather residue after degradation, and finally plant growth. So these are the main highlights of my research. So with this method, I was successful enough to isolate a keratinolytic bacteria named as Bacillus tropicus, which was able to degrade 40% of chicken feather and was efficient enough to support plant growth when compared to the control plant growth. Research articles I have published, and thank you so much for the patient listening. Thank you.
Okay, now moving on to regulatory affairs in biology, we have Divyani Chatterjee with her title being Organoid Technology, Pioneering Pathways in Biomedical Research.
Am I audible? Yeah. So, good evening, everyone. I'm Divyani Chatterjee. I'm doing my bachelor's in Manipal School of Life Sciences in Manipal. And today, I'm going to be presenting on the topic, Organoid Technology, Pioneering Pathways in Biomedical Research. Now, imagine this. I go up to someone and I tell them, each and every organ in your body can actually be created in the lab, and we can test things on it. A sane and sensible person would probably call me crazy. And you know, a decade and a half ago, they probably would have been right, but not any longer, because now we have organoids. So what are these organoids? They are three-dimensional tissue-engineered structures, which are mini organs, which are created in vitro and can be used for multiple testings. Now, the main thing of these organoids is that they can functionally mimic each and every organ, and also they can be used to observe all the developmental pathways in the lab. They are mostly derived from stem cells, and there are a variety of different types of organoids for different applications. So the applications extend into regenerative medicine, into personalized and precision medicine, into drug testing, and even into phylogenetic studies. The list is endless. Now the main uniqueness about the organoids is that it can be very easily integrated with artificial intelligence. Now why is this so? Well, they have good potential and tests can be done very easily on them. Also, where there is a requirement to generate a large amount of data and analyze data in a short duration of time, especially in drug testing and precision medicine, where we require repetitive tests, it is very, very useful to have this tool. Now, all of this technology, such a great, vast technology, so one quote comes to mind: with great power comes great responsibility. And thus, the ethical and regulatory landscape should be observed with a lot of care and concern. So the main thing about organoids is, since they are very new, the main ethical issues and regulatory issues are subjected to informed consents of the donors and also the established production guidelines of the organoids. But there are a lot of issues when it comes to market use and commercialability. So which include the cost capping. How much should these cost? Also, what about the ownership of the organoids and the intellectual property rights? Who owns them? What about protecting privacy and anonymity? What about if I get a disease if an organoid is implanted into me? Now, all of this requires research. And the main proposed solutions which we have come up with is that we already improve upon the consent forms which is present and also educate the donors, tell them exactly what they're getting into. Next is, we can also have extensive therapeutic and diagnostic testing so that we can understand what are the long-term effects of the organoids. Plus, research. This is definitely the last but not the least, because research unearths a gold mine of data which help us in the future. So having all of this together, we can hope to see that we can reduce animal testing and also have a society where organoids can actually cure every disease. Thank you. Have a nice day.
Thank you.
Thank you very much. Thank you.